This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1235, recorded on July 11th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's here in Western Mass. It says it's 82 Fahrenheit, but the humidity is 65%, and it really kind of feels sticky here. And yesterday we had a line of bad thunderstorms go through and poured down rain and did a lot of damage. Well, not a lot of damage. Did some damage, but we didn't get any. So today's a little nicer.
So yesterday, the TWiV with Daniel was episode 1234, the password in 90% of everyone's computers in the world. In fact, I just went to the bathroom and it's got a lock on it. And the password is
1234.
1234. Oh my gosh! Also joining
us,
and the
other
from,
10%, the password is password.
Yeah. Yes. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's 91 degrees and partly cloudy, so it's a pretty typical, maybe a little cooler than normal July day. And unless you've been living under a rock, you will know that the weather in Texas has not been great. We had, as we were recording a week ago, even though I didn't know it at the time because it was just on the front end of it, we were building up to a huge disaster, a real tragedy. There is a silver lining to that, which is that the local reservoirs that were getting really dry are now well up in volume. So it solved that problem, but I don't think it's worth the price. I'd rather have a drought.
We have three guests for you today. Special episode from Denver, Colorado, David Barton. Welcome to TWiV.
Hello, everyone.
And from Denver, Brian Kempf. Welcome.
Hello. I'll let you know that right now in Denver it's 85. We do have a chance of rain later today. Earlier this week we had our second 100-degree temperature of the summer, so not always looking forward to those heat days, but it is a dry heat, unlike Massachusetts and Texas.
In New York, but we don't get to 100.
Yeah.
We don't do 100 very often. Yeah. So both David and Brian are in Denver at the University of Colorado, and our third guest is from Fort Collins, Olve Peersen. Welcome, Olve.
Hey, welcome, Vincent. Great to be here. We have a similarly balmy 84/29 degrees up north. And
how far are you from Denver?
We are about 50 miles north, straight up the Colorado Front Range, as the mountains are called.
So I was there a couple of years.
Those clouds over my shoulder is up toward Olve's way.
It's a nice view behind you there. It's a good. Are you sure it's real? It's not like a fake backdrop?
That is what it looks like outside my window. And if you could look out, it's a bit hazy today, but those clouds are right over Rocky Mountain National Park.
Oh,
okay.
That's cool.
And Fort Collins is
adjacent to that.
That's the home of WWV, the National Institute of Standards time station.
Yeah, the transmitter is up here, and the origin, I think, is in Boulder.
Yeah.
So if we're really lucky, there'll be some good thunderstorms in the background for Dave here in a little bit.
So I was in Fort Collins a couple of years ago for a bat meeting. Did I see you there, Olve?
Yeah, you did.
You went to that meeting, right?
Yeah. Tony Schounts has a bat colony here on campus, on the west side of campus.
He brought me to see it. It was very cool.
It's pretty fascinating.
Very tiny bats, like about 4 inches long. What kind of bats are they? Are they Egyptian fruit bats or something like that? Do you know?
Fruit bats of some kind. I cannot remember exactly what it is.
Yeah. Well, if you like these programs where we talk about science, we give you real science, no lies, no misinformation. If you need that, if you want it, we also throw in some public health and epidemiology. Please help us to do this. We'd love your financial support. Microbe TV is a nonprofit in the U.S., so your donations would be federal U.S. tax deductible. You can go to microbe.tv/contribute for the various ways that you can help us out. Before we talk about...
Sometimes we might give misinformation, but
we correct it.
that's because we don't get it right all the time.
Yeah.
So misinformation is when you make a mistake, is that right?
Yes, misinformation is wrong. Disinformation is wrong on purpose.
Purpose, okay. So RFK Jr. is mostly disinformation, right?
He's a mix.
A mix.
And sometimes it's hard to tell whether he meant to lie or whether he actually believes what he's saying.
It's very difficult to tell. Because it's so outlandish sometimes that you have trouble thinking that anyone would believe that sort of thing, right? But there's always someone.
Yeah.
Let's find out where these individuals, how these individuals got to science. Is that an airplane?
That's a National Guard F-16 flying over the buildings here.
Oh, wow.
Probably a pair of them.
They usually come in groups of 3 when they take off and
they
fly
do threes,
over.
Right, yeah.
Oh, so you have a Air Force base there or something, like a National Guard base?
Yes.
Okay.
Up in this area, we get currently F-15s, but soon it'll be F-35s. They're usually in pairs.
Okay. Well, here we have fire engine sounds instead of fighter jets.
I can't do much better than a cat.
A cat.
Yeah, I got that too.
Now, let's find out a little of your histories. Let's start with you, David. Where are you from originally?
I was born and raised in Northwest Ohio, along the western basin of Lake Erie. So I grew up in the rural area. I learned biology in the creeks and the ditches, and growing up on a farm where you raise small animals like rabbits and chickens and pigs. And so all of my good biology education was before I got any formal education.
So that's why you decided to go into science, because it was all around you.
Yeah. I love biology, and so then you just got to figure out how you can make a living doing that.
Yes.
I had a similar experience. There was a creek that had a strong influence on me.
Yeah, I had that growing up in Maryland, out behind my grandparents' house. There was a little creek.
Are they back, or is it a new set taken off?
This is
Yeah. As Brian says, they come in groups, and so that might be the last 2 or 3 right there.
That might be the second flight. Yeah.
It's so unusual, it doesn't bother me. I can tell that it's a jet. So then when you went to college, you had already decided to do science?
Yeah, I was very interested in biology. I went to Ohio State for undergrad, and I kind of played the field there and tried to discern what I really liked. And after trying a botany course and a zoology course, I gravitated toward microbiology and got an undergrad degree in microbiology. So I'm a small biologist if you go with microbiology. And then, as you know, I trained in the Zwicky lab where, they imparted upon me a love of RNA and RNA viruses. And then, of course, after doing work with them on alpha viruses, I went to the Flanagan lab at the University of Florida, where Rich Condit was at one time, and we overlapped there for a while. And I studied picornaviruses and poliovirus in particular in the Flanagan lab. And so picorna fits into my good little small biology thing. I like small RNA viruses. I really yearn for things that are small and understandable. So I still am even though we live in this, systems biology world, I'm a reductionist at heart. I want them to be small and simple so I have a hope of understanding them.
So the Sawickis, that was in Toledo, is that right?
Yes.
That's
yes.
right.
Yes.
I was trying to remember.
So, Rich, did you remember David from?
Oh, of course.
Toledo? Oh, yeah.
Yeah, I remember lab meetings and that kind of stuff. Yeah, it was. How long were you at UF?
For 7 years, from 90 to 97.
Yeah, because I think we overlapped from 90 to 97. So I was there the whole time. I arrived at 90.
Yes. And so Penny Black was someone who got her PhD with you, but she did a rotation in the Flanagan lab.
That's right.
And she did a really critical experiment as it related to our cell-free replication reactions at the time. Right. She did the first guanidine reversal experiment when we, learned to work with these so-called pre-initiation RNA replication complexes back in the day.
Right. Yeah, Penny Black. Penny was awesome. Still is awesome somewhere in Kentucky.
So you showed that you could take polio RNA and put it into a cell extract, and out would come infectious viruses, right?
That's correct. You'd have
So
all
like-
the metabolic steps of replication in a little tube of cytoplasm.
And so the first time showing that a virus doesn't need a cell, right?
Yes.
Oh,
Yes,
okay.
life without cells. So you've got these RNA organisms replicating through RNA intermediates, and it sort of harkens back to the primordial RNA world.
But nobody has done that for any other virus since then, right?
Not really. People have played with rhinoviruses and EMCV and got them to work to a degree.
Mm.
So related viruses.
But
So when
nothing-
you
no
first
other-
did that experiment, was titering the lysate an afterthought, or did you go into it thinking, "Man, I wonder if we can make virus"?
Well, these were reported by Octermola in Eckhard Wimmer's lab. And at the same time, my initial project in Bert's lab was not going terribly well. So I adopted what they reported at meetings, and
Okay.
then they soon published it. And so
Right.
I adopted that experimental protocol. They
right.
had already validated it and talked about it at meetings. And so,
All right.
as you know, Bert Flanagan worked with purified polymerase, and as Olve does, of course, in my ongoing collaborations with him. And I like biology, and so the purified polymerase assays and the idea of reconstituting replication was okay, but really it was just enzyme assays. And so what was nice about the cell-free replication assay is it was biology. The virus would go through all of the natural steps of replication, and you could even in theory do in vitro evolution. We never pushed that, but they did those long ago with the Q beta systems.
I remember
Cool.
that many people had trouble doing those, and they always would call you and try and get you to troubleshoot for them, right?
There was a lot of lore of how you made a good versus a not so good cell extract, and
Yes.
I don't know that there was any. There's a bit of an art to it. Let's say I can attest to it. I think Brian, who did these for a long time, they're frustrating from time to time. They're not so easy that you can just read the recipe and make them work. But they do work when you get the good hands that make them work.
So, Brian, where are you from?
Originally, I'm from a small town in Wisconsin, right on Lake Michigan, called Manitowoc, Wisconsin. It's about 45-minute drive south of Green Bay, Wisconsin. So yes, I did grow up as a Green Bay Packer fan. I went to school at the University of Wisconsin at Stevens Point, one of the smaller four-year campuses, and I was a biology major there. During my time there, I had a summer internship, the SPUR program, the summer program of undergraduate research at the Medical College of Wisconsin in Milwaukee. And I worked on herpes viruses and immune regulators. So it was ISG 15 at the time. And while I was there was my first taste of virology. From there, I went to Colorado State University for my PhD and worked for Barry Beatty and Carol Blair on the cross virus. And then the day after I defended my thesis, I started working in Dave's lab. That was just over 22 years ago, and I've been here ever since.
Mm-hmm. So did you did you get interested in biology because it was around you, or was there something else?
So I come from a family of science. My dad was a junior high school science teacher, and my mom was a nurse. So our dinner conversations were always very scientific. My older brother also has a PhD in microbiology. He worked at Jet Propulsion's lab on the Mars rovers, planetary protection, and then he took a teaching job at University of Tennessee Martin. But he's a bacteriologist. My younger sister is a physical therapist. Obviously, you guys have met my wife on TWiV previously, so just science has been in my blood from the get-go.
All right. Did your brother read what is it? The Mars book by what's his face?
The Martian?
The Martian. Did he read The Martian?
I'm sure he has. Back,
I wonder what he thinks of it.
yeah, back when they were assembling the first two Mars rovers, my wife and I actually were out in Los Angeles visiting him, and we got to sign the guest book that they digitized and put onto a CD-ROM and loaded it onto I believe it was the second rover that was launched. So
Cool.
up there on Mars is my name on a CD-ROM unfortunately it's a dead rover now, but those rovers lasted a lot longer than anybody expected them to.
Nice.
Very cool.
And you should mention your wife's name.
My wife's name is Emily Travanti. She is the lab director at the Colorado State Public Health Laboratory. She was on TWiV in January of 2021 when they found the UK variant of SARS-CoV-2 here in Colorado, the first time detected in the United States.
Yeah.
Olve, where are you from?
I'm actually Norwegian. So going way back, born in Norway, and I came to the States with the whole family when I was 12 and landed in Kentucky, in Lexington, on a horse farm nonetheless. So I became cheap labor for my grandfather and spent a lot of time.
People say that Norway is such a great place to live, right? Like one of the highest rated places in the world. What'd you come here for?
Good question. No, at the time, my mother was widowed with 4 kids, actually, and married a guy who was American and decided we will make a go at moving. Why not? And I think it's turned out pretty well for all of us in the end, even though now I go back to Norway and say, going back might be kind of nice someday. We'll see.
So you came here as a young kid. How'd you get into science?
So I was always a little into... I think nature was one. I think especially in Norway, kids grow up and they're just outdoorsy people all the time. I was also into tinkering and building things. I was the kid who ripped apart my grandfather's watch and put it back together and got it to work. And so I really thinking, actually engineering when I went to college was kind of my primary lean. Happens to school I went to, Carnegie Mellon University. I was not quite good enough to make the cut for the EE program. They told me that as a high school senior, which I really appreciated. But I liked the school enough, I went. And I ended up saying, I'll do my biomedical engineering. In the end, did biological sciences, which was really a biochemistry, molecular biology program. There still is. Had a virology class, did not like it one bit. I will admit that. Went to graduate school at Yale in biophysics. I did like 3 years of undergraduate research, got into nuclear magnetic resonance research, protein structure, kept some of that going in graduate school, then came to Colorado for a postdoc in more biophysics in Boulder, the University of Colorado in Boulder. And then the second postdoc in Boulder with Steve Schultz, whose lab had actually solved a crystal structure of poliovirus polymerase, which was the first viral RDRP, RNA-dependent RNA polymerase structure solved. So I joined his lab working actually on a telomere binding protein, so DNA binding protein, but learned crystallography. And then started my own lab up here. And interestingly, this is the twist. I started my lab here at Colorado State doing chromatin telomere binding proteins. At the exact same time, Steve Schultz decided to leave research, and he went to teach at a Navajo tribal college out in Arizona. He had a really strong commitment to rural education from growing up in a small town in Minnesota. And so at that time, he suggested, why don't I pick up this polymerase project? Carla Kierkegaard and Ellie Ehrenfeld were also in this group, but they're strongly to suggest to do this. I was working in the lab with Ollie Richards, who had retired and was living in Boulder and wanted just free a lab to work in. And so he had worked with Ellie a lot. And so he was living in Boulder and basically in Steve's lab, not getting paid, but just wanted to stay in the lab. And Ollie and I came up and we crashed the ASV meeting here in Fort Collins in 2000. And I said, okay, I'll think about doing this. And so when I started my lab a couple of years later, we got into viral polymerase structures, and that took over my lab. You know, an enzyme that actually did something and did a virus replication was much more exciting than a static chromatin binding protein. And so we kind of switched into a viral replication protein lab.
Despite you not liking virology way back, huh?
Right. It's been about face, let me tell you.
Well, you just had to get to a small enough piece of the virus that you were interested.
Or maybe the engineering angle. You know, a lot of what I've done is solving structures, and I think about the polymerase as a molecular machine. Now all my engineering neurons are just firing. and once you get to the mechanism and the molecular things, how these things work.
So when were you at Yale?
I was at Yale from 88 to 94.
And that was your PhD?
That was PhD in molecular biophysics, and I was doing
What lab?
A guy named Steve Smith,
Okay.
who went on to Stony Brook afterwards. But
Okay.
it was solid state NMR on membrane.
So were
protein
you up
structure.
on the hill in like Klein Tower or something like that?
Next to Klein in Gibbs.
Okay.
Yeah, we were on the 6th floor of Gibbs, which was the afterthought floor where all the mechanicals were going to go.
Right.
And then they decided we need space. And so the elevator only went to 5. And we had to because the 6th floor was supposed to be all the mechanicals. And that building was raised, 5 or 6 years ago now, and it's been replaced with a very nice new facility.
All right.
All right, so let's talk about some science. We're going to throw out questions, and if we don't ask someone in particular, then any three of you can decide who's the best to answer it. But first, this one is for you, Olve, because we're going to be talking a lot about the RNA polymerase, the RNA-dependent RNA polymerase of poliovirus. So maybe you could give us a layperson's picture of what it looks like and how it works.
Okay, so how it looks is pretty easy. It's 500 amino acids long, so a chain polymer like that. I like to say it looks like a right hand. So if you hold up your right hand, you have the shape pretty well. It's a small kind of single subunit polymerase. So if you hold up your right hand and you put your thumb kind of to your index finger, and if you really want to twist your fingers and you put your ring finger now underneath your thumb, and I realize that hurts a little bit, you kind of have the shape of what this looks like. And the structure of it really has a palm domain, it has a thumb domain, and it has these fingers in that geometry. And then to do the replication...
That's the gang sign of picornavirologists.
There you go.
This is my low resolution version of Olve's high resolution.
Right. And given how hard that structure was to get, sometimes my middle finger extends a little bit. But it's actually really funny how hard it was to get that first structure, given how many more we've solved since my lab and other labs. And I think it was just... polio was a medically important one to go for, so that's what people went for. In hindsight, it was probably one of the hardest polymerases to work with.
Was the original structure from crystals?
The original structure was from Steve Schultz's lab, and it was a crystal structure. Everything was back in the day, so it was solved back in 97. It suffered what I call the unfortunate woodshop accident, which is to say all of the fingers were missing. There were just these bloody stumps. There's an interesting story there I may get to in a second. But
so do you do structures by cryo-EM now?
Now we are going into cryo-EM. A single polymerase is pretty small to get by cryo-EM at 50 kilodaltons, 500 amino acids. But we actually now have, from a Coxsackie variant, a really nice structure, looks like, with some RNA coming out in elongation complex, which just amazes me, the technology and how much easier it is to get.
So now this partially closed right hand, I mean, it's easy to envision an RNA molecule going through the thumb and finger. Is that how the RNA aligns?
Exactly not. So that was the early supposition. There's a big hole. We can put it through that big hole. But no. So what happens is, from the top of the finger, the template, the original copy of the RNA goes in, takes about a 90-degree turn, and comes out kind of along your hand, if you were to think about it. And then in the active site, which is in the palm domain, is the chemistry group that's there. And so the nucleotides are going to be incorporated, come in through that hole. So that hole is the NTP entry channel, we call it. And they wiggle their way in there and get into the active site and base pair with the RNA that's sitting there and make the new copy, which is then a duplex helix that comes out kind of toward you if you're holding your hand out in front of you. So that little opening, that hole, is basically the NTPs coming in, and the pyrophosphate afterwards leaving.
So the what comes out is a duplex, and what? How does that become single-stranded?
Okay. That is a very good question. Now, we really don't know. It happens external to the polymerase. but we don't have a firm handle on what does. We know that one polymerase can following a second polymerase following the first can strand separate and go through that duplex. The second one, so passively going through, it is not clear if there's a helicase that is dedicated to separating that. There is the 2C helicase in the coronaviruses, but whether it works during the RNA replication or the packaging of the virus particle is
and just for listeners who are not coming from virology, the context of this is poliovirus, all the coronaviruses as RNA viruses. They come in with a single strand RNA genome, which then has to be duplicated by making the opposite strand, and then that's copied additionally, and that's what this enzyme is doing. It's copying.
But the enzyme does do both strands.
Right.
I mean, same enzyme does both strands.
Right.
But you make you come in with a positive strand, you make some minus strand, then do you make an excess of positive strands?
Yes.
Okay.
So does the enzyme have a way of distinguishing so that it makes more positive strands than negative strands?
I'll let Dave can address, actually, probably that's more molecular virology. But let me say one more thing on the polymerase.
Which we know you don't like.
Kind of do the overview. Because this one pertains, this is an interesting little story on these polio polymerases. Really, they're the smallest replicative RNA polymerases. So you can go to flaviviruses and they get a little bigger on the polymerases and the genomes get a little bigger. You go to a coronavirus, it's got a 900 amino acid, twice as big of a polymerase. But in the core of all of these polymerase structures, you basically have the same thing we see in the polio polymerase, which is these bigger viruses with bigger genomes, decorate them with additional domains. And so when the chemistry happens, one of the things that these viruses do is polymerases in our cells will often recognize that nucleotide coming in and kind of bind it and recognize the right nucleotide and swing it over the active site where the chemistry is going to happen. And that active site is sitting there waiting for something to arrive to do the chemistry. The viral RNA-dependent RNA polymerase is really unique in that the new nucleotide comes in, and when it binds, then you structure the active site underneath of it. So at the end, all these polymerases look the same. You have a nucleotide sitting there ready for chemistry, and you have an active site that's structured. But the viral ones bind a nucleotide first and then structure the active sites. And the other ones bind a nucleotide and swing it over a pre-structured active site.
And if it's the wrong nucleotide, it doesn't swing it over? Is that how it works?
Well, in this case, it doesn't swing. Yeah, right. So in those polymerases, it swings over less efficiently, or it doesn't swing it over because it doesn't form the right interactions. But I think that's really important for these RNA viruses, because what it means, and this is work that Grace Campagnola in my lab worked out after we got the structure many years ago, is there's a really tiny molecular movement needed to kind of close the active site and trigger the chemistry. And there are a few amino acids that are important for doing that. But things are moving, by 1 angstrom, so 1.5 angstrom, so 1 times 10 to the 10th, a 10 billionth of a meter, a tiny little movement to trigger the chemistry. And I think it gives these viruses an amazing ability to evolve different speeds for how fast they go and fidelity, how much they recognize, how strictly they recognize the nucleotide being involved and can make mistakes because of it.
So how do you know that? Do you know, like, you have structures plus and minus nucleotide or something?
Yeah. So years ago, I had a very talented postdoc who solved the structure of the elongation complex of polio polymerase. So with an RNA, we basically put it on RNA, had it elongate to make a few nucleotides, and then stopped it. And it ended up being a very stable complex, which we did this by crystallography. And for the non-structural audience, in crystallography, you have to make a lot of material. You concentrate it very slowly, and you hope to form a crystal. And sometimes you get an aggregate and precipitate, but it's kind of analogous to a sugar crystal or a salt crystal, just a lot more complicated with a protein. And we're very fortunate to get crystals of polymerases, polio polymerase and Coxsackie polymerase and the rhinovirus polymerase. Once we had a technique, we were able to solve a lot of these structures. And what was amazing is, okay, we had the structure of RNA going through a polymerase as a stalled complex, but then we would throw in nucleotides like cytosine triphosphate, C, and it would actually do the chemistry in the crystal.
Ooh.
And then we would modify that a little bit, and some did chemistry and some didn't. And the protein would change its shape, and that's how we learned how the molecular machine was actually working to make this tiny movement to close the active site.
I would think that with that small a movement, it might take some convincing to believe that this is true.
Right. Yeah, it did. We had to do a little reviewer convincing too, but
Okay.
but I think that what was interesting was this totally different mechanism these viruses use, and I think it's a lot of why we don't have effective antivirals drugs for the polymerase inhibitors, because the movement is so small. It's very hard to get in the way when not very much is moving.
Mm-hmm.
And any drug you would bind would have to compete with high concentrations of these nucleotides in the cells, so it's just hard for them to get in.
So what's the resolution of these structures? Because it seems to me an angstrom is like less than the resolution of a lot of these structures, right?
Yeah, so the resolution for these was about 2.2 to 2.5 angstroms. And so the rule of thumb, the accuracy of knowing an atom position is about a tenth of that.
I see.
So we know an atom position to within a quarter.
All right. Okay.
So David, sorry.
Go ahead, Vincent. Yeah.
I was going to say, David, can you address this question of why you make so much more plus than minus?
I would start by saying it's not clearly delineated how you make more plus than minus, but some aspect of that involves both making these uridylated protein primers, VPG, PU, and those are clearly required for plus-strand RNA synthesis.
Mm-hmm.
And so you make a molar excess of these VPG, PUs, and somehow after you first make the minus strand, and you only make one minus strand for the incoming plus strand, that minus strand gets fixed into the replication complex, which is membrane anchored and has all kinds of protein-protein RNA interactions. That minus strand gets fixed into that membranous replication complex. And then these uridylated VPGs prime initiation of plus-stranded RNA synthesis over and over and over, because you make hundreds of these uridylated VPGs, and those get used stoichiometrically for one plus strand after another, presumably until it kind of runs out of gas. Overall, if you measure this asymmetry of minus strand synthesis to plus strand synthesis, it's about 50 to 1 under the conditions of cell-free replication reactions. It's reported to be 100 to 1 or more by people doing experiments in tissue culture cells, but somewhere in 50 to 100 to 200 to 1 ratios at the higher end. And so part of the magic of how you make an asymmetric amount of plus over minus is it's got any number of different mechanisms underlying that. One is fixing the minus strand in the membranous replication complex, and then reiteratively using these primers for plus strand RNA synthesis.
Okay, so there are essentially chemical differences in the way the minus and plus strands are being presented to the polymerase. The minus strand being held down, the plus strand having these caps attached.
Yes. How the minus strand is actually fixed in the replication complex, I don't think is well defined.
Okay.
Okay, so let's talk about recombination. So what is RNA recombination?
So recombination is when you take two different parental RNAs and you create a chimeric RNA by copying part of one and then switching from that template to another and copying another. And then that new genome is made from two different parental RNAs. And so then those are called recombinants. I like the theoretical concept of asexual and sexual replication, right? Asexual replication is, we were just talking about, minus strand, plus strand, is when you copy a full-length plus into a full-length minus and a full-length minus into a full-length plus. There's no genetic exchange. There's no chimeras formed. It's just one template copied into the other complementary strand and then back and forth. That's clearly an asexual replication cycle, which is well appreciated in the virology community. The idea of recombination being a sexual replication mechanism, I don't think is widely appreciated in the virology community. And of course, it's not absolutely true for all viruses, as Vince and the rest of you know, right? Minus strand viruses just don't recombine. They have different replication mechanisms, nucleocapsid wrapping up everything, and they don't really allow recombination. Plus strand RNA viruses, and in particular, picornaviruses that replicate often at mucosal surfaces, they love to recombine. And so you can get co-infections, you can get chimeric progeny out of each and every co-infection out of each and every human being for the most part. And so they love to switch, start making a minus strand, pause for sort of unknown reasons, come off of that first template, go find a related template and hybridize with that new template, and then tickle the polymerase the way the polymerase wants to be tickled, as Olve has taught us. And then they'll resume elongation. And you make these chimeric minus strands that then get copied into chimeric plus strands. And that happens readily in plus strand viruses, certainly very readily in, polio and non-polio enteroviruses.
So
definition of sexual reproduction because it's two
Sure.
genomes coming together.
Yeah, it's totally fine. Give you variation as you wouldn't.
Yeah.
Just
And so
copying.
recombination, I think, it's certainly pervasive across biology. You can get DNA recombination, RNA
Sure.
recombination, but this idea of putting it into the context of biology in general, right? Asexual and sexual replication, most people on the planet would understand that. So recombination is just a form of sexual replication.
And so mutation and recombination are the two main ways that diversity arises in organisms, right? And so they're
Yes.
both happening. So it's funny that you mentioned the negative strand viruses don't recombine. I have my students in virology look at Contagion, the movie, and tell me 3 things that are wrong with it. And this year,
They didn't show the toilet paper shortage. That's
this year, a student came up with something, said, "Professor, you said minus strand viruses don't recombine, and supposedly this is a recombinant of 2 negative strand viruses." I said, "Oh my God, you're right.
good one.
Isn't that a great one?"
Artistic license. Then yes, even when you teach this, you miss the errors that are right in front of you from time to time. This
Yes.
is why students are wonderful. Students are
Yep.
Students are a good way to bounce ideas off, and then eventually they'll see something that you've just missed for whatever reason.
So
I
does
have a couple.
recombinant?
I have a couple of recombination questions. First of
Sure.
all, can you give me some idea of how efficient this is? I don't even know how to express that, but maybe you've got some numbers.
So Brian and of course other people in the field have created their recombination assays. So Brian created a recombination assay in our lab where he engineered a lethal 3 amino acid deletion into the active site of the polymerase. So if you do Olve's polymerase and you go right into the palm where the GDD activation domain is, Brian deleted the GDD. So it doesn't have the aspartates to orient the magnesium to do the chemistry that Olve was just saying. So that's a lethal mutation in an otherwise full-length RNA. And then we have a subgenomic replicon, which came out of the Flanagan lab way back in the day, and we still work with that. It has a large capsid deletion. And so you can take a full-length virus with a debilitated polymerase, which is lethal, and you can take a subgenomic replicon that doesn't make capsids, but it does make all the replication machinery. You can put those into cells individually, and you get 0 virus back. You put them into cells together, and the replicon will do the RNA replication. And of course, it can then recombine with the full-length molecule that has the capsid genes, and you can measure the frequency of how often it recombines.
Okay.
And so if you transfect full-length infectious RNA into murine cells, you can get 10 to the 6 PFU per mil out of the murine cells. If you do these 2 lethal mutations individually, you get 0. So you get a millionfold signal to noise here. If you do the 2 RNAs together, you get about 10 to the 3rd, 10 to the 4th PFU per mil. So that's about 1 out of 1,000, 1 out of 100 from what you would get out of just full-length infectious virus. So by that measure, recombination is, 1% to 0.1% of your average replication rounds.
Reasonably efficient.
Relatively efficient.
And do you ever get more than one crossover?
You typically, by the mechanisms that we've outlined, you get 1 crossover at a time, but those can happen independently over and over and over.
So you
Right,
can,
and then those recombined genomes could potentially be templates for another
Absolutely.
round.
Absolutely. And you know, the biology that we've sort of unraveled in recent years with Olve's help is that when you replicate by asexual mechanisms, the polymerase can throw in errors, and those will make the virus less and less fit. And if you have enough mutations accumulating due to multiple rounds of asexual replication, if the mutation burden becomes so great, the viruses become unviable. By recombination at some frequency, which the viruses have obviously evolved to do, the recombination just sort of randomly expunges lethal mutations, and then you go back to the wild type. So there's a balance in every infection where you have a lot of asexual replication and then a little bit of sexual replication, and the sexual replication helps expunge the mistakes that the polymerase occasionally pours in.
That's
And this
interesting. It's kind of like reverse transcription. The genome of retroviruses is diploid, and so the same thing happens. The polymerase is going back and forth and reconstructing an infectious genome, right?
Yes, and of course, with the retroviruses, they're diploid in the virion. With the picornaviruses, as soon as you start making new plus strand progeny, there's plenty of other molecules to recombine with. But they actually share ancient coevolutionary origins, right? So the RDRPs and the reverse transcriptases, people in the evolutionary world like to argue about which ones arose first, but their structure function, as Olve described the structure of the RNA-dependent RNA polymerase, the reverse transcriptases have very similar structure function to mediate recombination.
Sorry,
now,
Al, go ahead.
if you modulate I'm not sure exactly how to do this experiment, but I think it's in one of your papers. If you try to increase the amount of recombination or decrease the amount of recombination, can you see where the threshold is, where you get this error catastrophe that asexual reproduction accumulates too many errors, and see how close to that the virus normally is?
We can manipulate it to a degree and then measure the impact on error catastrophe. We can't prevent it completely and still have a viable virus.
Right.
And so we can debilitate recombination with this so-called L420A alanine substitution mutation, which is in this so-called extended primer grip right adjacent to the active site that Olve described. And so that amino acid substitution just downstream from the active site inhibits homologous recombination in Bryan's recombination assay by about 50 to 100-fold. And so, but that doesn't eliminate all recombination. It knocks it down by 50 to 100-fold. And then when you measure ribavirin-induced error catastrophe, those viruses are much more sensitive to error catastrophe, where this antiviral drug, which exacerbates the incorporation of mutations into the virus, makes the viruses non-viable. And so those mutant viruses are much more sensitive to error catastrophe than the wild-type virus.
So the wild-type virus with its normal rate of error versus recombination is kind of close to going over the line into error catastrophe, and the recombination is kind of saving it. Mm-hmm. So there's a balance that's evolved here.
Right. And as Olve mentioned in his lab, he's sort of characterized polymerases from across the picornavirus world. Some of them are naturally faster, some of them are naturally slower. So some of them are closer to this error threshold and some of them are farther away from this error threshold. To me, it's interesting from a biological selection point of view. Why would some viruses choose to be faster than others? But the fire the danger you're dealing with is if you fly too close to the sun, aka you go too fast, you have to deal with the negative consequences of error catastrophe.
So among the three polioviruses, do they have differences in where they are with respect to error catastrophe?
Well, in reality, the polymerase across the three polioviruses is an arbitrary thing because they share them with each other in these recombination groups. So there is no such thing as a polio 1, 2, or 3 polymerase. These recombination groups that we've just published on, they share their polymerases with the other members of the recombination group. So there is no defined polio polymerase per se.
Okay. So, I mean, typically you have to have co-infection of two different viruses to get recombination, although I can imagine in a quasi-species you wouldn't even have to have different viruses. But let's put that aside for now. So
it rescues itself in that context.
It rescues itself, right. So, but you can also have co-infection with different... So how far apart among the picornas can recombination occur?
So what we've just published is if the polymerases are about 6% different at the amino acid level, they recombine with each other. And that's sort of one of these hallmarks of the viruses in a recombination group, is that the polymerase gene gets shared from one parental RNA to the other. And of course, it can go backwards. And so in those recombination groups out in nature, all of those polymerases are about 94% identity at the amino acid level or higher. If you get farther away, they don't recombine efficiently in nature, and then they kind of go off on their own evolutionary path. And so the paper we just published, we took what the current ICTV defines as enteroviruses, and we subdivided that into 4 recombination groups, where in nature, those viruses that we've defined actually recombine with each other, and it sort of defies the capsid-based phylogeny of the ICTV.
So there are multiple A, B, C, D, et cetera. So they will only recombine within the letter?
Within the recombination group.
Within C, for example.
In C. So there's 4 recombination groups, 1, 2, 3, and 4. And polio type 1, 2, and 3 are in a recombination group with about a dozen other viruses. So Coxsackie virus A, 13, 20, 21, 24. I can't remember them all off the top of my head.
All right, but what you're saying is the enterovirus classification does not necessarily predict recombination groups.
That's correct. All of the classic ICTV classification, especially at the species level, has some let's say arguable flaws. And those those taxonomic systems are based largely on capsid gene sequence, right? So serotype, right? And so what we're defining is more of an RNA polymerase dependent categorization system, and they're both valid in their own regard. But all enteroviruses do not recombine with each other. There's these subsets that we've defined in this recent publication.
Will an enterovirus recombine with a cardiovirus, for example?
So the recombination groups that we've defined are where they recombine with each other all the time in nature. That doesn't preclude having more promiscuity, if you will, in who you choose as a related enough versus not related enough. So there's always more promiscuous recombination that sort of punctuates geologic evolution. What we're talking about with these recombination groups is something that happens, week in, week out within any co-infections. The more, it's uncommon, if not undocumented, to see polio recombined with species A viruses or species B viruses, for instance. And in fact, what it really combines with out in nature is this little subset of species C viruses that we've defined.
I like how this comes back to the fundamental biological concept of species, which is that
how do you know who's in a species,
who
is not related?
They can interbreed.
Who do you want to reproduce with is as simple as it gets.
Right.
Yeah, that's the original definition of a species, right?
I'm anticipating a long letter from Jens coming in sometime soon.
Yeah. I don't want to pick a fight with the taxonomists, but they have a difficult job, and I don't want to poke them any more than necessary.
So
when we look at sequences, when we take a co-infection of poliovirus and Coxsackie A21 and purify the virus, the RNA, extract it, and send it for sequencing, deep sequencing. And just the various experimental systems that we use, we do see that these viruses, they will recombine. They will start with the coronavirus, either the polio or the Coxsackie A21 RNA, and then it will have a portion of human genome RNA. It's got messenger RNA on it as well. So while, yes, it's able to recombine with a lot of different things, it comes down to the functionality of the end product. And if that end product doesn't produce a functional virus, it's a dead end in recombination.
Right.
So I think this is approaching a question I have, which is there nucleic acid homology involved mechanistically in the recombination? Do you have to match up bases?
Yeah.
And so there is, according to what we've just reported, and it's this really beautiful small region adjacent to the active site. And this extended primer grip, which includes the place where we can make mutations to inhibit recombination, there's about 8 bases of double-stranded RNA just downstream from the polymerase until the double-stranded RNA goes away from the footprint of the polymerase. It's really the first 4 or 5 bases of double-stranded RNA. So about 1 or 2 codons if you're thinking across open reading frames. And so the polymerase is only using 5 or 6 base pairs immediately adjacent to the active site to define what's a homologous crossover site. That leaves lots of room for promiscuity. And so these protein RNA interactions that Olve has helped us define, in particular in this latest paper, are what tip the balance in favor of the virus being able to distinguish what they think is more related and less related. But it's not absolute, right? If it's only 1 or 2 codons of base pairing, that leaves lots of room for promiscuity, which was what Brian was saying. You can see a lot of evidence of promiscuity where you get non-homologous recombination, but this little bit of protein RNA interaction adjacent to the active site is enough to tip the balance into the ability of the virus to pick in a favorable way homologous sites over non-homologous sites. Okay. And so, we're going to try to get one more cycle out of the R01 here, and we're going to define the bejesus out of what is tolerated and what's not tolerated in terms of misbase pairing in this region of the polymerase.
So you get these non-homologous events, but the system skews toward homologous because of this.
Yes. So Olve created this beautiful little assay with his purified polymerases where we can feed it a template where it's either homologous in this region and it takes it and runs, or if you have a mismatch in the second or third base pair away from the active site, this polymerase elongation complex that Olve got these beautiful atomic structures from, it falls right apart. And so it goes from being stable for, 90 minutes to 2 or 3 hours. And then if you put a mismatch in the second or third double-stranded piece adjacent to the active site, it'll lower the stability dramatically of those elongation complexes. And then if you do the mismatch plus an L420A mutation, they go down to a stability of 5 minutes or something like that. I hope I'm not exaggerating.
30 seconds.
30 seconds. I wasn't even close to exaggerating.
So
this little region of the polymerase, right, where it's got template and duplex, it goes from being super stable to being ridiculously unstable if you have a mismatch and then add on top of that this mutation that inhibits recombination. And so that's the essence of the paper, of the data we just published. So it's those protein RNA interactions right at the heart of the polymerase that determine who is and is not a suitable partner for recombination.
So once again, mechanistically, I may be getting ahead of ourselves here, or maybe you can't even answer this, but I'm having trouble visualizing how this happens. So I have to take an elongation complex, and I have to, in my mind, I'm seeing moving the polymerase and the nascent strand detaching from the template and moving to another template, physically moving. Am I thinking about that right? And if
yes,
so,
I think by our point of view, it's really the nascent products that move. They leave the polymerase and the old template behind.
Oh.
And then it has to recruit, then finds a new template, and then somehow recruits a new polymerase. But those details are, let's say, a bit of hand waving.
Okay.
Olve might be able to describe this in a different molecular way where everybody doesn't have to move quite so much. Because, yes.
No, I think the end result that you have 2 hybrids coming together is there, and template switching is the phrase that's for it. I'm not being the virologist necessarily by training. I also think about it maybe as a primer swapping, that maybe a polymerase for some reason aborts but stays associated with a template, and then a new RNA wiggles its way into that active site and is or is not fully homologous. And one question is, how much homology do you need to extend that RNA on the other template that's already there?
The core
And
of it,
I'm kind of
the
envisioning
core of it is that the nascent products are moving from one template to another.
Right. Okay.
And then the polymerase is either already there, or you need a new polymerase, or who knows?
So I don't get this either. So we got a plus strand. Isn't it threaded into the polymerase so that it just can't fall out? Physically,
Yes.
right?
It is, as a template strand, it is threaded. It is somewhat labeled and come in and out, but you can't take it out and leave the product strand kind of behind. Molecularly, that would just be really difficult.
Yeah.
It's like the template comes in first and then the product comes on top. So if you're going to think about the switch that way in the middle of the genome, it makes more sense that something stops a polymerase, you get an abortive product that floats around the cell and then comes back, and a new template will come into that.
So that product leaves
Yeah.
the polymerase, and
Mhm.
then it gets a different polymerase and a different template to complete
Right.
it.
Yeah.
And
So,
so,
so naturally, of course, these elongation complexes where you've got a template and a nascent product are enormously processive, right? They go from one end to the other end, except from when they don't, which is this ratio between, how much recombination versus asexual. The asexuals, at 0.1%, but at 1 out of 1,000 times, those nascent products are going to come off, and they're going to go look for somebody new.
And when those nascent products, when say a plus strand comes off and it's partial and it's drifting around, it's got homology with minus strands that are there being used as templates, right? Does that
Yes.
help it
Mhm.
bind to some existing site and get itself extended?
That's the thinking, and the question is how much homology do you need right there?
Right.
But from a mechanistic point of view, the recombination happens during minus strand synthesis. So it's nascent minus strands coming off of the plus strand and finding a new plus strand. And, as Vince would say, because I actually like his language from his textbook, he articulates why recombination is during minus strand synthesis, but not during plus strand synthesis, because there's always an excess of plus strands around. There's never an excess of minus strands around. So all of the literature going back to Carla Kierkegaard's experiments from some time ago suggests that recombination occurs during minus strand synthesis, not during plus strand synthesis. And that's a function of how many what are free templates available, and there's always an excess of plus strands.
So it would be the minus strand that comes off the polymerase and then possibly hybridizes to some plus strand?
It looks for anything that's related, and yes,
Right.
Here we go.
Right.
okay, so as you know, OPV2 was modified by Andino and guy in England,
McAdam.
what's his name?
McAdam.
McAdam, and a bunch of others. They spent billions of dollars to make NOPV2. It went through trials. It's released. Many hundreds of millions of kids get it. And in the end, it still causes poliomyelitis, just like OPV2. Maybe it's slightly better. But one of the problems is they introduced mutations throughout the genome, including in the polymerase. And one of them was to reduce recombination. But it wasn't L, whatever your number is, L420.
It was not L420.
So why didn't they use L420?
I wasn't in the room when they decided, but when you're making an attenuated vaccine, you have the trouble of you can't make it too attenuated.
Yeah.
I in the end have no idea why they chose the mutations they did and didn't. In the end, it's very hard to engineer mutations in a polymerase that will inhibit recombination, especially when you consider you only have one of the two partners to be able to mutagenize, right? The other partner out in nature is not being mutagenized, and they have functional polymerases. And so this idea that you can mutagenize the polymerase to inhibit recombination is a challenged idea.
But the idea that, all right, so whatever, I don't know what position they introduced to knock down. They tried to make it have higher fidelity and less recombination.
Sure.
But it's not zero ever, right? Recombination is not going to be zero, and so that allows you to recombine.
Exactly. And again, you're only mutating one of the two partners, and they're both making protein in the infected cell. So it's largely a challenging thing to conceive that you can inhibit recombination.
Challenging is a nice word, right?
Yes.
For this.
Yeah.
So we know what it was an enterovirus C that it recombines with. Does this fit into your grouping of recombination partners?
Yes, sir.
It does. Okay.
So all of the circulating vaccine-derived polioviruses that we're aware of, and we keep curating the system and looking through them as they are reported, they all recombine with a subset of group C enteroviruses, which by our classification are recombination group 4 viruses. So it's a subset of viruses that all have a polymerase that's 94% similar to all of the other polymerases in that recombination group. So Coxsackie 13, 20, 21, 24. It's a small but notable subset of polio and related group C enteroviruses.
And those are fairly common circulating viruses.
In particular parts of the world, yes.
Yeah.
Okay.
Yeah. So this for listeners who just came in recently, what the big problem with the oral poliovirus vaccine is that it can mutate back to a virulent form and cause paralytic polio. And the NOPV was an effort to try to fix that, and that's what we're talking about, that it didn't work because it can still recombine with these other picornaviruses that are out in nature and get its pathogenicity back.
Yeah. So outside of the polymerase mutations, they had some very clever mutations into that novel OPV2, right? They debilitated the natural Cree, moved one up to the other end, and so then it would require a double recombination event, and then they layered on that iris mutation by which then disallowed the virus from reverting to virulence by point mutation and selection. So all of that was cleverly done and is still true. That said, in nature, these viruses recombine. It's a double recombination event, and they're largely back to, wild type virus.
So that's what I forget who said earlier. You can have more than one recombination event. That's what happened with these viruses.
Yes. It would be 2 sequential recombination events.
And those recombinants can cause poliomyelitis, right?
Yes.
And they can also circulate in unvaccinated populations.
Yes.
Or people who have got IPV, where the gut is fully susceptible, right?
Yeah, they can circulate silently and then, of course, cause disease in people who are not vaccinated.
Yeah.
So just requiring that it recombine twice is not a high enough barrier in a virus that's going to be put in and undergo multiple rounds of replication in the gut and have all these opportunities.
Yeah.
To
The real weakness is that the OPVs, whether they be the Sabin strains or the novel OPV2, is that they transmit from person to person.
Yeah.
And during that process, it's inevitable that they'll co-infect someone with a related group C enterovirus and then be able to recombine.
So when they were constructing and testing NOPV2, did they not try co-infections with enterovirus Cs to look at
they did. And so, McAdam and Raul Andino published a paper in Cell Host and Microbe in 2020, give or take. And they did tissue culture experiments where they did co-infections and they measured recombination. They showed in those tissue culture experiments recombination in the replication genes, right? The P2, P3, it was mostly P3 recombination events, but they did not see recombination that swapped out the 5' UTR mutations, which were the key attenuating mutations. So in tissue culture, they didn't see evidence of the double recombination that's evident in nature. Now, that said, the selective pressure in tissue culture cells is much lower than it is in your gut. And when a virus is, hanging on by a thread and it wants to survive, it'll recombine twice if it has to. In a tissue culture cell, the competition's not really quite as acute.
So this is all about fitness, right? It's
yes.
It's the selection pressure is fitness, and that's much greater in the gut than a tissue culture plate.
Yes.
So the title of one of their papers was, engineering OPV2 to prevent reversion to neurovirulence. Now, Andy McAdam argues that this is not reversion because the 5' end is not changed; it's just removed and replaced with a more fit 5' end. How do you view that?
Well, again, this goes back to the biological context of asexual and sexual replication, right? It's this bedrock of biology is that you want to pass along your genetic material to future generations. And so these viruses have been doing this forever. It's what they do. They when you pick a partner to recombine with, it's clearly a fit partner, because you're swapping out what you already have for something your new partner has something that's better. And so the idea of this, idea of asexual and sexual replication, I think is really important in these concepts, because it's easy to understand. When you reproduce sexually, you want to swap out your inferior cistron for your partner's superior cistron. And that's, of course, what happens with novel OPV2. When you have a purposely debilitated gene segment, it can go replace that with wild type versions out in nature.
Right.
And I guess the, it's really a terminology question. It's not a reversion because it's not the original sequence back in. It didn't reconstruct itself as type 2 poliovirus. It reconstructed itself as something that's functionally equivalent because it picked up, the N from some other virus.
It's a phenotypic reversion.
It's a phenotypic reversion. It is not a genotypic reversion, which is not a distinction that matters if your kid is the one in the wheelchair.
Yeah, of course.
Right.
Of course.
Right. It's still an infectious virus that came out of it.
Yes, it's still virulent.
So, I mean, in retrospect, is there anything they could have done to avoid that and to realize that recombination would have negated all the work they did?
I've obviously followed, polio eradication for a long time and their efforts to design viruses. You'll remember at one time there were two candidates. There was the macadam andino version, and then out of the CDC, they were doing codon deoptimization in the capsid genes. What they're trying to eradicate, of course, they're trying to eradicate the capsid genes. They're not trying to eradicate the non-capsid genes. Mm-hmm. And so with that in mind, it would make sense to attenuate the capsid genes. The macadam andino thing, they attenuated outside the capsid genes. And so then, of course, recombination just replaces all of that, and you're still left with wild-type capsid genes. So from a conceptual point of view, it would make sense, if you're trying to eradicate the capsid genes, design your attenuating mutations into the capsid genes, and then there's nothing useful that can swap those out in nature.
So you're saying we should have used the codon deoptimized
Well, they did run that through, phase 1 and or 2 trials, and they decided that it didn't meet their immunity measurements when they did the Latin American trials. And the challenge with vaccines is it's a series of binary decisions, and every decision can be good or bad, and so it's a real challenge. And so they, of course, had two candidates, and then they narrowed it to the one, etc. And here we are.
Would recombination have negated the deoptimization?
Codon deoptimization, it wouldn't. In theory, you'd have to find polio capsid genes to recombine with. And as you might know, there's not a lot of polio capsid genes out in nature anymore.
Yeah,
I like this idea that the eradication, we're not eradicating the virus, we're eradicating the capsid. That's an interesting way to put it.
These organisms are very odd. The capsid genes evolve separately from the rest of the genome. It's just the way they are.
And what makes it a poliovirus is the capsid, really.
Exactly, by terminology. That's just a human moniker.
Actually, what makes it a virus is the capsid, right?
Yes.
Because there are lots of...
It builds their spaceship to move from cell to cell.
Yes, because there were lots of RNA replicators in the world before viruses, but they weren't viruses because they didn't have capsids.
Right. But it's also a bit of a passport to certain cell types for what kind of cells to infect, what the capsid binds to.
Yeah. So the reason NOPV2 was made was because OPV2 was a problem, right? It caused the vast majority of vaccine-derived poliomyelitis. So does that have something to do with the RNA polymerase? In other words, does it make more mistakes and therefore it's more likely to revert in the 5' end?
In terms of Sabin strains?
Just number 2, because that made the most number of vaccine-associated paralytic cases compared to 1 in 3, right? And that's why they made NOPV2 to get around this type 2 problem.
Again, the polymerase, because of recombination happening all the time, the polymerase comes and goes, right? And so there is no one polio polymerase. There's a recombination group 4 polymerase, which gets shared from polio to polio and polio to non-polio and back and forth again. And so when people classically defined differences in virulence between type 1 and 2 and 3 polioviruses, there was never, I don't think there was much of an appreciation of these recombination events, but the non-capsid genes were different from, one strain of type 1 polio to another.
Sure.
And whether, non-capsid genes impact virulence is certainly not questionable. They certainly do, but whether they impact virulence in a measurable way that we understand in nature, I think is an open question, because it was just never addressed very systematically.
So basically, no one has taken OPV2 polymerase and studied that.
Head-to-head in a lab-based assay, that could tell us, be informative about what the virus is doing. That's a pretty tall order in some ways.
All right.
Just hard to get at replicating. Even recombination.
So we don't really know what makes OPV2 so good at reverting to virulence.
No, not really. There was a nice paper from Korotkov, or I'm going to screw up the name, but 2017, give or take. And they looked at what was intertypic recombination from polio type 1 to 2 to 3 and back and forth again. So when you did trivalent vaccine, the polio strains would recombine with each other. And they would, in particular, recombine out the polymerase. There was a pattern to it. I don't remember the pattern off the top of my head, but type 2 might have been the one where it had the polymerase that was more likely to be swapped out, and then who swapped out who amongst type 1, 2, and 3, right? And so that was how the study was set up, was who picked up a type 1 polymerase versus who lost a type 2 polymerase. And so one could pursue that to just compare, which of the 3 Sabin polymerases were more fit or less fit, because one would get swapped out by the other.
Right. It wasn't that the type 2 was the weakest of the 3 polymerases?
I think that was the case, but I don't remember clearly.
Yeah.
Weakest in what sense?
The weakest in that it would be replaced
most readily
in the three horse race.
So in fact, there's no polio 1, 2, and 3. They're all recombinants, right? And we just happen to have isolated one in someone's lab where there's polio 1, 2, and 3. That's a recombinant that they happen to have. And
the serotype is defined by the capsid genes. The rest of it is non-capsid genes, and those non-capsid genes come and go based on recombination in nature.
So it's like phages, which are mosaics, right, of all kinds of other phages. Or maybe you don't know that, but there's for any phage, which you give a name to, there's a whole range of recombinants that you can get. Yeah, so it's the same idea. So you talked about the Cree before. I want to ask you a Cree question. Maybe you should explain first what a Cree is. We've talked about it a little.
Please.
So Cree is an acronym for cis replication element, and it can be either more general or more specific. In the more specific nature, there's a particular cis replication element in the open reading frame of picornaviruses. It can be in the 2C gene of some viruses or in the capsid genes of other viruses, but it's a template with, it's a little stem loop RNA structure with 2 or 3 A's in a loop, and the polymerase uses those A's to add uridine to the VPG primer. So it goes from VPG protein, where it's got a tyrosine hydroxyl, and it adds 1 U, it slips back, it adds another U, and then it drops off that VPG PU and does it all over again. So in this case, there's a cis replication element that makes the primer for plus strand synthesis, which may also be the primer for minus strand synthesis.
So in NOPV2, they moved Cree from, I think, 2C up to the 5'
That's
coding
correct.
region. Why did they do that?
That was to make the virus more resistant to recombining out the attenuating mutation that they engineered into the iris, right? So they engineered this very clever mutation into the iris to attenuate the virus so it can't revert by point mutation in selection. And then they move the Cree to help protect that from a simple recombination event. So they debilitated the natural Cree and put an unnatural Cree up there next to that structure that they wanted to protect. And so the long and short of it, you'd have to replace the natural Cree with a functional one before you can recombine out the upstream one. So that was designed very cleverly. But of course, that's exactly what the virus does. These recombinants that have been described by the really beautiful molecular epidemiology that they do, right? They find viruses in nature. They find viruses that are associated with paralytic polio. They sequence them. In this case, they derive from the vaccine. But you can see that all of the very clever mutations that were engineered into the novel OPV2 are almost surgically removed by recombination events.
So they could not outsmart it, basically.
Not so far.
So the first recombination event put in a Cree,
so
right?
one could argue about the sequence, but you could argue that one recombination restores the natural Cree, and then the second recombination event,
Right.
Fixes the front end. And Nora Chapman did really beautiful experiments back in the day where she would mutate one Cree and try to put in another Cree, and you can't. And she would have two Crees, and those were really bad. So you can't have two wild type Crees at the same time. Was part of her work, and so the virus would have to do these sequential recombination events in short order, because if you don't have a Cree, you're really debilitated. If you have two wild type Crees, that's not ideal either.
So
But the usual biological solution and the usual viral solution is you just keep throwing bodies against it. I mean, we're talking about billions of replication cycles.
Yeah, there's lots of shots on goals
when somebody takes this vaccine, and so it's going to happen. Okay,
Mass action. Yeah.
So the reason I ask about Cree, and maybe I got this wrong, but I thought you published a paper once using subgenomic replicons where you showed the movement of the Cree had little effect. I guess that's the basis for moving it to the 5'9".
Yeah, that was actually other people that did a lot of the moving the Cree around stuff.
Okay.
We would bash the Cree and knock it out and study it whether it's there or not there, but we didn't do a ton of moving it around. Others did that sort of work.
One of the things that's running through my mind is this is a great example for you lab leak freaks out there. Great example of humans trying to construct a virus that will behave a certain way and putting it out in nature and
humans with years of work and a lot of funding.
Right.
Yeah.
Yeah, so there should be a lot of humility in the field, that's for sure.
Yeah.
There should be, but I think saying you could make a virus that doesn't revert is a lack of humility, in my opinion, right? Because I think the thing is a failure. It's no better. Gates spent $4 billion on it. It's no better, and yet WHO continues to push it as the solution.
Yeah, I would say it's incrementally better because it does presumably prevent vaccine-associated paralletic polio by point mutation and reversion, but it can still revert in the long run.
Recombine.
So,
Yeah.
So recently there was an outbreak of circulating vaccine-derived type 2 polio in Papua New Guinea. The viruses are recombinants, as we've discussed, but enteroviruses C are not thought to circulate there. So what do you think is going on?
I would have to look at the reported sequences. I love when the CDC and others publish these complete genomes, and I get people who have better bioinformatic skills than myself to look at them quickly because we keep checking to see if there's any deviation from what we're suggesting to be true, is that there's this defined subset of viruses that they do recombine with. So I would expect that there's some group C enterovirus that they found, whether humans knew it was there or not.
So it's a matter of surveillance,
Yeah. Just because you don't see it doesn't mean it's not there.
So you probably know what we think about using OPV for eradication. What are your thoughts on that?
Yeah, obviously eradication's been a long and winding road. It's a goal that's so far been difficult to achieve for so many different reasons, right? The socioeconomic, the political, the scientific reasons that we draw attention to. There's been naysayers of eradication for a long time. I've never been terribly outspoken, but I have my own opinions. I think one of the real problems was this idea that Wimmer interjected into the things when he showed by synthetic biology you can just recreate polio, and so eradication ain't what it used to be if you can just recreate things, and so it's not durable over time. It doesn't seem to be a milestone that would change public health in a dramatic way. You're going to want to maintain immunity against polio both before and after eradication if it's ever achieved. So it seems to be a lot of effort for a milestone that doesn't necessarily have durable impact on humanity in perpetuity.
Well, part of the problem is that many countries are using IPV, and like the U.S. and many European countries, and their polioviruses circulate readily in those
Yes,
populations.
yes. So the ability to achieve worldwide mucosal immunity that's durable for some period of time such that you can eradicate polio, no one's found the way to do that just yet. Yes, we've all learned over and over again that mucosal immunity is not very durable, and polio knows this. Polio has been going from person to person for a very long time, and it's very difficult to block every chain of transmission all at the same time.
So you think we need a new polio vaccine?
It wouldn't be a terrible idea.
What kind of vaccine should we have? What any ideas?
If you could get nucleic acid free vaccines that provoke sustained mucosal immunity that maybe you could use more regularly, that wouldn't be a terrible idea if you want to achieve things like eradication.
And you don't want nucleic acid, so there's no recombination going.
Right.
Correct, unless you're going to put some debilitating mutation in those capsid genes.
It's also got to be cheap to make, and it's also got to be able to avoid having to use a cold chain, especially
And it has to be deliverable orally.
Deliverable orally, yeah, because needles are
Logistics.
a problem. Yeah, the logistics of delivery are, well, most people, when they think about vaccine and vaccine delivery, might not think about those logistics. Those logistics play a significant part in how well a vaccine campaign or an eradication strategy are going to work.
Well, logistics is the whole reason OPV is still in use.
Yeah, so economic reasons, all of this.
If we could give everybody in the world a shot, we wouldn't be having this conversation because everybody would get IPV.
Right.
Yeah, and of course the world's evolving in that direction. IPV is becoming more widespread,
Right.
and it's it's a very good vaccine in many ways, but it's not going to be something that leads to eradication per se.
Well, the WHO plan is to transition to IPV as part of routine healthcare of children, right? Which is different from a focused eradication program. And in fact, as you know, the Gates Foundation is going to sunset in 2045. And after that, they only talk about maintenance of no more talk about eradication. It's all about maintenance of low levels of poliomyelitis. So I think
the disease, not the virus. Yeah.
The disease, because I don't think you can get rid of poliovirus
No.
unless you
I
think
agree with
it's
that.
going to burn itself out. You think that would happen?
At some level, these are terms they used in 1988, long before I started studying poliovirus. But they used the word in some of these articles that it was inevitable that polio would be eradicated. I don't necessarily disagree with that. It's just a matter of time, and you can go with control. IPV is great for control. It's not going to be great for eradication. But I still sort of believe in my heart that if the world continues to evolve in the right directions, that eradication's inevitable. It's just not inevitable in the near term. This idea that you can force it to happen quickly and then that's the end of all the intervention that needs to occur, I think is wrong. It's wrong-minded. You need to achieve and maintain eradication, which means you need to maintain immunity indefinitely, in perpetuity. And so if you have a strategy that's designed to give people whatever vaccine works best for their subpopulation based on cost and et cetera, that can happen over a long view. It's not something that should be forced in the next 3 years and then we don't immunize anymore. That's not the best approach.
Removing
I'm hung up here on the idea of a vaccine that does not contain nucleic acid and induces robust mucosal immunity. I'm trying to think of whether there are examples of that. Human papillomavirus vaccine, that's almost a sterilizing immunization, right? Which I would think would be a mucosal immunity, but I'm half making that up. Do you know of any examples?
Well,
Is this a realistic...
HPV is just protein, right?
Yeah, right.
But it's injected. It's not even delivered mucosally.
Mhm.
So I don't know.
There are mucosal surfaces in the body where IgG leaks out. So, deep in the lungs, you
Yes,
can get
sure.
IgG, in mucosal surfaces of the rest of the reproductive tract can be protected by IgG that leaches out. So in that case, a protein vaccine that gives systemic immunity can give a degree of mucosal immunity as well, if you believe in this idea that IgG leaches out and provides protection at those mucosal surfaces.
Because flu and RSV and COVID vaccines are all injected, and you get mucosal resistance. So but we don't want injections.
But to your point,
right?
Rich, it's true.
No.
There are no sort of great vaccines that are shown to be protein only that induce good durable mucosal immunity. Right. That would be a good basic science breakthrough. Somehow, how do you go about having an adjuvant or whatever it is that gives you durable mucosal immunity?
And how do you get that through the digestive tract?
Yeah, and you got to solve Brian's problem as well. Can't be injected.
Yeah.
Can't have a cold chain.
Well, this is what research is for, right? And as you know, after June of this year, you can't work on polio anywhere unless you have a polio essential facility. So you're just dampening the ability to innovate and make new vaccines, unfortunately.
Yes. This has been something you've brought up before that I do endorse. You can't get new innovative approaches if you tell everyone that you have the tools you need to complete the job, but you can't complete the job. And so if you argue that there's no market for the new tools that you clearly need, then it's hard to get the innovation that's necessary down the road.
I mean, that's why WHO is saying this will work, this new NOPV. In fact, they want to do NOPV 1, 2, and 3 now, right?
Yeah, but they want to base it off of they're basically, from my understanding, the plan is that they're going to swap out the capsids, so use the backbone of NOBV2 and just swap out the capsid proteins. And so we've already discussed that OPV2, the polymerase, is a problem and is readily going to get recombined out. Another problem is with every dose of OPV, be it 1, 3, bivalent, or the new novel OPV2 or whatever combination that they use, for every dose that they put out there, they are potentially seeding a new outbreak.
Yeah.
And that's where I have concerns with the eradication strategy, is that every time you use a virus that is attenuated and it has the ability to either revert through point mutations like the original Sabin strains, or has the ability to recombine very quickly like the novel OPV2 does, you are seeding new outbreaks. And so the editorial that was just published by Chumakov and Plotkin has in there that you're fighting fire with fire. Mm-hmm. And that's a great quote, because yes, while you are trying to fight polio, you're fighting it with a live polio virus. And that doesn't make a whole lot of sense.
It's unique to polio because we have these viruses in us that will recombine with, and Albert Sabin had no clue about that, right? But the yellow fever vaccine, the measles, mumps, rubella, they don't recombine with anything. Apparently, there's not virus. Well, of course, yellow fever they're all plus strand viruses, so they should, but there's nothing for them to recombine with.
Right.
So,
the 17D, the yellow fever vaccine, has a large deletion in it, which, as far as I know, hasn't ever been recombined back to make that vaccine transmissible.
But people don't have...
Enteroviruses are just more abundant in diversity, and these mucosal surfaces are just a playground for enteroviruses. So there's always more than one there, quite frequently.
Yeah, and when I've made lots of mutant viruses over the 22 years that I've been in Dave's lab, and I can tell you that poliovirus is incredibly resilient. When I was designing the recombination assay back in 15 years ago when I came up with the idea, and the first few trials that the first few mutants that I made were just simple alanine substitutions of the GDD in 3D polymerase, and put those into tissue culture cells, transfected them, and within 24, 48 hours, the cells were dead. And it's like, what happened? And so while you might think it's a debilitating mutation, while you're still having it, while it's debilitated, you haven't killed it. And it's able to revert through point mutations or recombination. In this case, because it was the only transfected RNA in, it reverted through mutations, through point mutations. So again, a lot of the tricks that you might try to throw at it to make it a more stable vaccine, or put these creative ways in and be like, okay, these mutations to make it a higher fidelity, or these mutations to affect its ability to recombine, the virus quickly gets away from those.
It's hard to prevent evolution.
Yes. And then
That's
the other thing that
good one.
Dave and I have been talking about, and I keep bringing this up as we have these discussions, is that let's say we do wipe out poliovirus, type 1, type 2, type 3. You've created an ecological vacuum. And if you go back to Aristotle's saying that nature abhors a vacuum, will one of those other polymerase group 4, the recombination group 4 viruses, evolve to fill that ecological niche that you created by removing those 3 polioviruses?
Sure. Who knows? Plenty of enteros that can cause paralysis out there,
Yes.
right?
Mm-hmm.
So is there anything we missed, gentlemen, that you would like to interject before we end here? We got everything?
Sounds
Okay.
like we covered a lot of ground.
Yes.
So
we
I
did.
have one more question for each of you. So David, if you had not been a scientist, what would you have done with your life?
Oh my goodness! I think when I was younger, of course, you already heard my love of nature. If I could make a good living being a forester, I would just go, hang out in the forest here, outside the Rocky Mountains here. But yeah, something out in nature.
Yeah.
And so,
Cool. How about you, Brian?
I remember I always wanted to be a chef if I wasn't a scientist. And so, yeah, I probably would have ended up as a chef somewhere.
Yeah, that'd be fun. Although I met a waiter once in a restaurant, and he used to be a chef, and he said, "You make more money as a waiter."
Yeah, yeah.
Yep, I've heard that. Yes.
But I do like the idea of nurturing the soul through food. There's just something very peaceful and meaningful about that.
Also, cooking is like science, right?
Yes,
Yeah.
that's what I was thinking.
Except you can taste the product.
Yeah, you can. Olve, what would you?
This is precise.
What would you do, Olve?
Other than the engineering thing I get behind, honestly, I probably something along the lines of carpentry. I think I like to make stuff, to see the product that
You're
you have.
still an engineer.
Right, kind of. But maybe a little more outdoors to go with it. I think I'd shy off of straight up farming. But
All right, that is TWiV 1235. You can find the show notes at microbe.tv/TWiV. If you want to send us a question or a comment or a pick of the week, you can send them to microbe.tv, TWiV at microbe.tv. And as I said before, if you enjoy these programs, we'd love to have your financial support. Microbe.tv/contribute. Our guests today from University of Colorado, David Barton. Thank you, David.
thank you all.
And also from the University of Colorado, Brian Kempf. Thanks a lot.
Thank you for having me.
We now have the Kempf family covered here on TWiV.
Kempf and Travanti, yes.
From Colorado State University, Olve Peersen. Thank you, Olve.
Yes. Great to be here.
I'd like that. Putting the index finger under the thumb, right?
Yeah,
the whole.
If it hurts a little, you're doing it right.
Oh, that's very cool. I have to remember that.
So,
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time and great to talk to you guys.
I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.